Many conversations about cloning end up being debates about whether cloning is permissible or not. But not everything about cloning is up for debate. So what I'm going to do in this short video is give you enough information about cells and microbiology and cloning so that you could understand where we can ask ethical questions when it comes to cloning. And then with the questions, you can begin to answer them for yourselves. The whole conversation starts with cells, because cells make up every living organism, and those cells contain DNA, and the DNA contains genetic information. And the genetic information, or genomes, which are the collection of those genes, include all the information about the organism genetically speaking. So not necessarily if the organism is going to be tall or short, but whether it's likely the organism would be tall or short, whether the organism can fly or can swim and can breathe underwater or can see in the dark. Your genetic information tells you a lot, but not everything about the organism which would grow or has grown. Cloning is when you take a biological entity and you produce a genetically identical copy. So we have cloned cats, and the clone, the thing that resulted by copying a cat, did not look like the original cat that was copied. So we now know that cloning genetic information does not mean getting all of the observable characteristics, just the genetic information. You can also see this when you're looking at twins. Twins don't necessarily look or act exactly the same, but they have exactly similar genetic information. So when we're talking about whether cloning is permissible, we're saying, what are you allowed to do with genetic information? Are you allowed to duplicate it? And maybe when are you allowed to duplicate it, or why? Maybe you need a particular reason, and then you could duplicate it. Well, not all the time is this a question of ethics, because some cloning takes place naturally. So that's obviously not up for debate. It happens on its own. For example, a cell replicating into another cell, replicating into another cell. That's how bacteria grows. Bacteria are single-celled organisms that reproduce by cloning. Also, some plants, they have asexual reproduction. That's also, like bacteria, natural cloning. Also, in many mammals, eggs split into two, and now you have two fetuses growing. That is a third example of natural cloning. So when it comes to talking about cloning in the classroom, we're not debating every example of a genetic information copied into a new, right? So that's not what we're talking about. When we're talking about cloning in a classroom, we're talking about artificial cloning. We're talking about extracting genetic information from a biological entity and forcing it to be a new thing by copying it, by artificially creating a second biological entity, or maybe cell, maybe organ, maybe tissue, maybe whole entity. So, one very popular process by which we would clone artificially is called somatic cell nuclear transfer. Essentially, you are taking somatic cells, like for example, skin tissue, and you are going to take all of the DNA out, and then you're going to put a new DNA cluster in, and then let that thing grow. So that is one way to clone, and we'll talk more about that with reproductive cloning. So one thing I wanted to talk about are the three different and major ways to do artificial cloning. And one is reproductive cloning, but there's also gene therapy or therapeutic cloning. There's also gene cloning. So gene cloning is when you're just copying the genes or a sequence, a part of the DNA. Therapeutic cloning is when you are producing the embryonic stem cells, which are super important, and we'll get into that next. A third way or reason or process by which you clone is when you are making entire entities through reproductive cloning. Genetic therapy is that middle one, therapeutic cloning. Genetic therapy is a process by which you are going to fix mutated genes, replace mutated genes, or use genes to train a body how to react to foreign cells, for example, to boost the immune system, for example. So gene therapy is something you can do with embryonic stem cells. How can you do that? Well, when you have an embryo that's growing, it's going to make cells. And when it has about 100 cells at day 5 or so, you can go in there and remove the cells that the embryo is producing. Those cells will be at that stage of blastocyst stem cells. Stem cells are super cool because those building blocks of every human organism can become any building block. So the stem cells that you have around day 5 of an embryo, they can become the lungs of someone, the heart of someone, the blood of someone, the feet of someone. Stem cells can become almost any cell that you want them to become. So we can harvest the stem cells, thereby destroying the embryo, and then we can use those stem cells in embryonic stem cell research and in gene therapy, which is a therapeutic process by which we take some produced embryonic stem cells and then use them to fix somebody else's mutated genes. Reproductive cloning is very different because it has a different purpose, but the process is the same. You're going to remove the mature somatic cell from the animal you want to copy. You're going to remove all of the DNA containing nuclei, part of some animal that would serve as the host, and then you're going to add the somatic cell that you want to copy into it so that the egg now has the DNA information of whatever you're trying to copy. And then when you allow that egg to grow in a test tube, it will, if successful, start to grow into whatever you are trying to create. And when it's mature enough, you can implant that into a womb of a surrogate mother, and then nature just takes its course. So you would be able to reproduce an entire sheep, for example, from a small piece of DNA, including cell that you insert into the host. And this is when things get very interesting. We know that genes carry information about whether some organism is going to be healthy or unhealthy or have mental disabilities or have this or that ability, biologically speaking. And one of those things we know is that there are disorders or abnormalities or diseases that are carried through one's genes. There's a lot of different kinds of disorders, but we know that genes cause them or carry them. So what if we could take life, create it, stop it at the point where there's a ton of stem cells, take those cells and replace it in the DNA sequence where there is a mutation leading to one of these genetic disorders? Can we do that? Yes. Should we do that? Now ethics is getting involved. Now, another thing that's happening is we're picking and choosing what kind of life we're saying has value. And we're saying that people with some kinds of diseases have mutations in their genes that make that organism less valuable somehow. And that explains why we're trying to replace the DNA sequence portion that includes that genetic information so that we can get rid of those structures. Well, you're leading towards something called eugenics, whereby you figure out what you think are desirable or undesirable genetics or desirable, undesirable characteristics that are passed on through genetics, and then you deny reproduction to those with undesirable genetics, only allowing the reproduction for the desirable genetics. And in doing so, you are improving, so to speak, based on your standard of what's valuable and what's not, the future generation. Now, we already do this when it comes to fruits, for example. You take the seeds of your favorite tomato and you make sure that you plant those so that you get more of that kind of tomato. And if there's a terrible tomato, well, don't plant the seeds and you no longer have that tomato. We do this also with animals. We'll take the strongest cattle, for example, and we'll breed them, thereby picking and choosing the reproduction of what we have called desirable traits. That's the essential idea of eugenics. And that also allows us to ask a ton of questions like, are we allowed to do whatever we want based on what we value? Do we have the right values? Is there an objectively healthy DNA sequence to every species that allows us to look at a genetic code and say that would be undesirable or abnormal or a mutation? What's counted as a genetic mutation and what's not? Okay, we have cloned many animals, especially mammals. We've cloned cows and sheep and cats and deer and rabbits. And recently there's been some claims that researchers have cloned humans, especially out of South Korea, but with lack of proof. We are going to, as of right now, say that no human being has ever been successfully cloned. But we have cloned other animals, other mammals. By the way, the United States FDA has approved as safe your consumption of eating the meat of a clone or drinking the milk of a clone. Fun fact. So here are all of the questions that we can end with based on what I've said before. Is cloning humans, but also just life in general? Is it safe? A lot of the times what happens is that medical scientists are trying to ensure that some biological entity does not have some mutated gene, but then they get the mutated gene anyway. So we don't know perfectly well what we're doing. Is it safe to try? And let's say it is safe. Is it moral? But also we can ask if it's moral about specific kinds of cloning. For example, is it moral for us to create life and then destroy it because we just wanted to learn about it? Is it permissible? Is it moral? Is it ethical for us to create life just to extract the stem cells and save other people and destroy that life? If you think, for example, on the abortion debate that you are pro-life or pro-fetus, you think that a growing fetus has a right to live. Well, that would make it very hard for you to say that a scientist is allowed to see life growing, steal all of the stem cells, and then destroy the embryo. But also, if we were to clone somebody and use the copied genetic information to give them offspring, then does that make them the parent and the twin? What does that do to the parent-child relationship? But also, what kind of value are we placing on life in general if we are able to create it willy-nilly for research and then destroy it willy-nilly? So what are we saying about human life if we are living in a time where we are willing to create a growing fetus of a human just to destroy it for the sake of science or for the sake of saving other people? That's very utilitarian.